I would like to, first of all, I would like to thank organizing committee, especially Dr. Ruzo, for kind invitation from here. I'm very pleased to be here and with you. As you know, global environment and agricultural inputs influence the soil and growth environment. As a scientist, we have to, our duty, we solve the problem. One of the prospects of the role of plant growth promoting rhizobacter strain reduce application rates mineral fertilizer in barley. This study are part of our project's results. I will try to summarize some of the results in this presentation. In this project, supported by the European Union F6 project system, and our members was Australia, Belgium, France, Germany, Chile, Israel, Brazil, United Kingdom, and Turkey, whose main goal is to develop and improve understanding of specific rhizobacteria already identified and studied to some extent by partners, with a good potential as plant growth promoting, as called at the same time biofertilizer. REBAC aims to reduce the high inputs of the nitrogen and phosphorus chemical fertilizer commonly used in the Arabic cultivation. The crimes of the REBAC is that wheat or barley inoculate containing PGPR improved for enhanced plant growth promotion will enable reduction inputs of the chemical fertilizer while maintaining yields and protein content of the grain at levels acceptable for the relevant markets. Therefore, PGPR can contribute to improving the local and global environment and reducing the dependence on non-renewable research, while still permitting cereal growers to supply grain with quality and price acceptable to consumer. As you know, nitrogen and phosphorus is as known to essential nutrition for the plant growth and development. Intensive farming practice that achieve high yield require chemical fertilizers and which are not only costly but may also create environmental problems. The extensive use of chemical fertilizer in agriculture currently under debate due to environmental concern and for consumer health. So for this purpose, we produced a biofertilizer. We can be used different kind of application and crop dependent of the crop application. One of them is capsulating model is produced for the biologic fertilizer application in the soil and soil surface. Biologic fertilizer, when applied seed or plant surface or soil and directly applied in the plant root area. The first picture is showing that biologic fertilizer in powder types, we can mix it before the seed sowing in the field. You can apply it, and after that you can sowing in the field. And the second picture is showing that if you want to use spray for foliar fertilizer, if you want, you can apply. You can apply the foliar fertilizer and fertigation system. In the last case, as you know, it is very extensive in the agriculture and it is increasing use irrigation and fertilizer efficiency. The third one is soil application. Some crops and some soil condition it needs to apply the before the and during the seed sowing time. You can apply it on granule form or the spraying form. The third one is directly applied in plant root area on the capsulating form. This form is releasing dependent on the time and sustain the plant nutrition during the plant period. The sophisticated application is plane or helicopter application. Sometimes it needs. Maybe it is very use expensive, but sometimes you have to apply this kind of application. For example, this tea plantation area, Black Sea region, now its application is with helicopter application, because the area and land area is very deep and very sensitive for the tillage, and any application for this purpose we can apply in this kind of application. How can you use this PGPR in the agriculture aspect? One of them is plant growth promotion, biological fertilizer is one of them, and biocontrol agents. And the third, restoration of wastelands and soil bioremediation and stability on productivity of desert soils. PGPR has got a lot of efficiency, and we can use in the agriculture direct affecting, first of all, nitrogen fixation, and second, solubilization of insoluble phosphorus. It's very important in our country. Consequencing of iron production of the siderophores and lowering of ethylene concentration. Ethylene concentration is very important, especially in stress condition. As you know, global warming is getting increasing, and we now feeling the results in nowadays. And plants is, when plant is getting stress, we releasing ethylene in the root area, and root length and root development is restriction. But you can apply in this kind of fertilizer, you can alleviate this kind of problem. And production of phytohormones such as axin, cytokinin, and gibberellins is also very important for the plant growth period. Indirect effects is antibiotic production, depletion of iron from the rhizosphere, synthetic antifungal metabolites, production of fungal cell wall releasing enzymes, competition for the site on root, induced systemic resistance. As you know, it is very important for sustainable plant growth. You have to occur the nutrient balance. In generally, when you apply the mineral fertilizer or organic fertilizer in the soil, if your fertilizer efficiency is very lower, you cannot obtain the enough growth and enough crop production. For this purpose, we use in some chemical or biological application for the plant root area. One of them is nitrate leaching or phosphate leaching is possible in rainy area and over irrigation period. And the second one is phosphorus problem, especially in our country. As you know, is lime content is very huge. So when we apply the phosphorus in the soil, about 1%, only 1% or 20% maybe plant is uptaken from the soil, the phosphorus. The rest of the phosphorus is accumulating the rhizosphere area and fixed form. Total amount is getting increase, but plant available part is very small degree in the soil. When you apply the plant PGPR in the soil, we increase in the available plant phosphorus in the soil. The other method is the solubilization of the rock phosphor, other minerals in water and liquid form. We have tested already, and we have obtained patent from the international. They applied this different kind of PGPR in this medium, rock and the other phosphorus form easily dissolve of the solution. But this effect sometimes is maybe mixing, and it is not related in the soil condition, because soil condition has got a tampon capacity, so this result can be changed. And the calcium carbonate at the same time can be solved, applied in the PGPR application. And we can use at the same time bioremediation and phytoremediation. We can be using PGPR for this purpose. As you know, there are a lot of microorganism located in the plant root area, beneficial microorganism amount is very important because this microorganism is producing some of the useful materials for the increasing plant nutrition. One of them is auxin and cytokinin and gibberellins. It is at the same time supported stress condition and increasing plant uptake. The important matter, the other section, is the root elongation. Root elongation is very important in the heavy soil and undesirable condition. When you apply the PGPR, it is increasing root elongation and density in the root area. What is the aim of our study? To efficiency of plant growth promotion, PGPR in barley, to investigate effects of the alone on combination with the nitrogen fixation and phosphorus solubilizing PGPR strains on efficiency as a biofertilizer, and to test efficiency of PGPR inoculation in barley, low blend and plateau, and cold high blend in depends on plant growth, nutrient uptake, and grain yield of the barley. We selected two locations. One of them is Çoruh Valley is low level land, and the Erzurum is high level land, about 2,000 meters high level from the sea level, and about 34 rows as to give a 180 kilogram per hectare barley seed, and treatment is as shown is the controlled OC 142 M3 SP 245 and etc. This picture is shown that it's seeding time in this study and growing period. What was the result? The soil, two sides, soil chemical and some physical properties as shown that its organic matter level is moderate and it is enough for the plant production, and phosphorus, potassium, calcium, magnesium is some of the sufficient or insufficient level. When you compare the treatments according to application treatment, nitrogen, full dose of nitrogen fertilizer, about 4.02 kilogram ton hectare the amount of the crop yield, and mixing application is 3.85 ton hectare, it's the crop. The second year result is similar to the first year, and when you evaluate the total average yield, first application control treatment straw yield 6.84 ton hectare, grain yield 2.73 ton hectare, and 9.52 ton hectare. When you apply in half of those nitrogen application, in straw yield about 8 ton hectare, grain yield 3.7 ton hectare, and total yield 11.8 hectare. But full dose of nitrogen application, straw yield is 9.36 and grain yield 4.7 ton hectare, and total biomass 14.43 ton hectares. What was the mix of the PGPR application? About 8.32 and straw yield and grain yield is 3.9 ton hectare, and total biomass 11.25 ton hectare. Similar results in obtained low level land, and the total yield is accurate about as the same. I want to take your attention this picture, when you apply the half of dose and full dose of nitrogen fertilizer, the plant is getting increase, because you apply application of nitrogen is at the same time feeding of the wild plant and increasing some wild problem. When you compare to increasing ratio, according to grain yield, about 49% is increasing when you apply half full dose of nitrogen, and when you apply half of those nitrogen application is 30%, and mixed PGPR application is above the half of nitrogen doses. And at the same, with similar result, you get obtained this straw yield. This full dose of nitrogen is increasing to about 36% according to control, but mixed application is over than the half of nitrogen doses. The last one is total biomass, and total biomass, as is shown, that mixed application is over than the half of those nitrogen and nearest the full dose of nitrogen application. And the field, second field application. What was the plant quality and nutrient contents application of the PGPR? Nitrogen application is the highest the level in full dose of nitrogen application, but phosphorus and the others element in generally is very high level of the Vasile entry application. For the grain content, when you evaluate the mixed application is very important efficiency was obtained from the mixed application, and straw nutrient contents is the same. When you compare the control treatments and the other application, what was the difference, the application of the nutrition uptakes? Mixed application and full dose of nitrogen application about 37 and 43% increasing of the nitrogen application, and grain the same 73 and 81%, and the straw yield about 96 and 81% is increasing nitrogen application. Phosphorus is the difference from the application treatment. Vasile entry application is the most effective than the others application, about 32 and 20% is increasing ratio, and the other is very huge effectiveness of the phosphorus application. Potassium and at the same in Vasile is the most, and sulfur mixed application is the best, and iron and the micronutrient is very efficiency mixing, and Vasile entry application is very high level we get obtained this result. And the last of the presentation, these topics in enzyme activity, because PGPR efficiency it is related to dehydrogenase and urease efficiency for the nitrogen content of the plant, and alkaline phosphatase and acid phosphatase activity is related to plant phosphorus level of the plant. When you, it's shown that application of PGPR is increasing dehydrogenase and urease enzyme activity. At the same time, plant nutrient and nitrogen content is similar to urease enzyme activity. The second one is phosphorus. Plant phosphorus content is related to alkaline phosphates enzyme activity, and our results indicated that microbial inoculation of the seeds with Nitrogen fixation and phosphorus solubilizing PGPR strains alone and in combination may subside costly and fertilizer in barley production, even in cold, highland and lowland areas, and provide plant phosphor requirement from the soil, phosphor exchangeable and moderately available forms reserves via increasing the phosphor stability. In view of environmental pollution in case of excessive use of fertilizers due to high cost in the production of nitrogen and phosphor fertilizer, bacteria tested in our study may well be studied alone or combination achieve sustainable and ecological agriculture production in the region. Our results also indicated that alone or combination inoculation with nitrogen fixation and phosphorus solubilization PGPR strains could increase mineral concentration and accumulation also. This study was supported view that inoculation with PGPR has some potential to serve as a means of to reduce higher through the enhanced mineral concentration and accumulation in the grain. The last presentation is showed that in Turkey, Finland, United Kingdom, and Israel. After 4 years period, we discussed the in-field study, and we advertised in local and international advertisement in media. It is the best organism is selected in our country, biofertilizer, and about half of the nitrogen fertilizer reduced in United Kingdom. United Kingdom now try to accept this microorganism from our country. They used about 250 kilogram per hectare nitrogen in their land, but after used our microorganism, about half of those nitrogen they don't use at the same amount production. This shown that Ribat, if you want to any detail for the study results, we can visit in Ribat in the internet, and we can easily show this study result. Thank you very much for your attention.
Thank you.